Hi, this is Dr. Neela Bakore, and in the previous segment we have discussed photosynthesis and how the plants, normal plants, perform photosynthesis using C3 pathway. Because of C3 pathway, as the enzyme required rubisco, its action or activities change during conditions like higher concentration of oxygen and higher temperature. It undergoes oxidation, or it helps in oxidation reactions, which results in photorespiration. So there are some plants which have adapted themselves to overcome this problem of photorespiration. Those plants, they perform a different cycle which is called C4 pathway, or it is also known as Hatch-Slack pathway, named after this scientist. C3 pathway is known as C3 because the first stable compound is a 3-carbon compound. Here it is known as C4 because the first stable compound is a C4 compound. It is the first stable compound, and that's why it is known as C4 cycle. Hatch-Slack, named after the scientist. The plants which show this kind of adaptation, or the examples of plants which show C4 pathway, include sugarcane, maize, sorghum, amaranthus, chenopodium, etc. These are the plants which show C4 pathway. Now, what exactly are the changes or adaptations in C4 plants by which they are able to avoid the loss due to photorespiration? Those changes are mainly two categories, we can say. One, they have Krenz anatomy. They show Kranz anatomy. In Kranz anatomy, the bundle sheath cells are arranged in a wreath-like manner. Wreath-like is circular arrangement of anything, is known as wreath. So here the bundle sheath cells, they are thick-walled, compactly packed, that means without any intercellular spaces. So there would be a wreath-like circular arrangement of bundle sheath cells. This is known as Kranz anatomy. The second important change or adaptation which is found in C4 plants is that they have dimorphic chloroplasts. Di means two and morph means form. So there are two types of chloroplasts which are found in them. One is granule and second are agranule. Granule means the chloroplasts which have grana in them. Grana are those stacks of thylakoids which are present in these chloroplasts. So if these grana are present, then the chloroplast will be termed as granule, and if grana are absent, then the chloroplast will have only some stroma lamellae and all other enzymes and everything in the stroma part. So granule and agranule. Granule chloroplasts are present in mesophyll cells. Whereas agranule chloroplasts are present in bundle sheath cells. Now, how have these 2 points helped in overcoming the problem of photorespiration? Let us talk about few things of photorespiration. in photorespiration the problem was that instead of binding with carbon dioxide, RuBP, that is rubulose biphosphate, binds with oxygen, and this is because of the changed behavior of rubisco. Normally this rubisco acts as a carboxylase, that means it helps in addition of carbon dioxide to RuBP where C3 cycle takes place. Now in this condition it is acting as an oxygenase. When is this behavior changing? The behavior changes when oxygen concentration is more, and 2, when the temperature is high. Temperature is high. In these two conditions, rubisco will start acting as oxygenase instead of carboxylase. So in this, the entire process, carbon dioxide is released. So basically, photorespiration results in release of carbon, which is fixed by photosynthesis. Now we have to think of these 2 points, how C4 plants have taken care of these two conditions. Let us come back to this bundle sheath. Now if we simply draw a leaf structure, maybe say this is the epidermis and here would be the mesophyll cells and the bundle sheath, wreath like arrangement of bundle sheath. So two things have been done. There are two types of chloroplast. Mesophyll cells have granule and bundle sheath cells have agranule. Granule chloroplast means these are the places, the thylakoids, where chlorophyll pigment is present. It will absorb light, or we can say light reaction takes place here. Wherever light reaction takes place, that is the same place where oxygen is released due to photolysis of water. So where will be oxygen released? At this place. Oxygen will be released in mesophyll. And agranule chloroplasts are present in the bundle sheath. Agranule means there is no chlorophyll, so here dark reaction will take place. So site of light reaction is mesophyll, whereas site of dark reaction is bundle sheath. That means oxygen will be released in mesophyll and rubisco will act in bundle sheath. So we have avoided a situation where the oxygen concentration will be higher. So what have these plants done? They have changed the sites of light reaction and dark reaction. Light reaction in mesophyll cells, dark reaction in bundle sheath cells. Light reaction will result in release of the oxygen as a byproduct, and in bundle sheath cells the rubisco, this enzyme, will act. So now if oxygen is not available, its concentration is low, it automatically will act as a carboxylase enzyme. So one problem taken care of. The second problem was of temperature. Now if photosynthesis takes place in these upper cells, mesophyll cells, these cells are closest to the surface, so temperature is going to be higher. If the site is shifted into deeper cells, as you go down and down into the deeper tissue, the temperature is going to be lower. So here the site of C3 cycle has been shifted from superficial mesophyll cells to deeper bundle sheath cells. So the temperature in the deeper cells will be lower. So by having these two things, Kranz anatomy and dimorphic chloroplast, C4 plants have totally avoided the problems caused due to photorespiration. Now let us take the exact process how this C4 cycle takes place. To understand how the cycle takes place, let us draw two cells. This upper cell which we are drawing is the mesophyll cell. And one more cell which is the bundle sheath cell. Now what happens? Through the epidermis, a gaseous exchange through stomata will take place. So now carbon dioxide comes in through stomata. This carbon dioxide is accepted by a 3-carbon compound called phosphoenol pyruvate. Let us write down what PEP stands for. It is phosphoenol pyruvate. It is a 3-carbon compound which is going to accept carbon dioxide. And the first compound which is formed here is oxaloacetic acid. So let us write OAA. It stands for oxaloacetic acid. It is a C4 compound. So first stable compound is oxaloacetic acid, and that's why the cycle is named as C4 cycle. The enzyme which acts here is PEPCO. PEPCO, P-E-P, PEPCO, it is called PEP, that is phosphoenol pyruvate carboxylase. It is called phosphoenol pyruvate carboxylase. And this PEPCO acts as carboxylase in all conditions. So there is no issue whether the temperature is higher or oxygen concentration is more. It will always act as carboxylase. So that problem which was due to Rubisco has been taken care of. Now this oxaloacetic acid is converted into, and let us write, it is a 4-carbon compound. It is converted into malate. For conversion of this malate, oxaloacetate into malate, reduction is required. So NADPH2 will get converted into NADP. There is one more possible reaction which can take place here, and the reaction is, let us write it on this side, that oxaloacetate can change into aspartate. Instead of malate, it can also form aspartate. Only change will be, one is same, NADPH2, that is reduction of acetate into aspartate, and one amino group is required. So amino will get added, then it will change into aspartate, and if it is simple reduction, it will change into malate. Whatever is formed, whether malate or aspartate, they will diffuse into bundle sheath cells. So in mesophyll cell, what has happened? Phosphoenol pyruvate has accepted carbon dioxide to form a 4-carbon compound, which is the first stable compound here, and that's why the cycle is called C4 cycle. It changes into malate by reduction, and this malate diffuses into bundle sheath. Now what we have here is malate. Again, it can be aspartate also. So malate, which is also a 4-carbon compound, dissociates into a 3-carbon compound, and 1 carbon is released in the form of carbon dioxide. So this 3-carbon compound is pyruvic acid or pyruvate. So this Pa is pyruvate, which is a 3-carbon compound. So now carbon dioxide is released. In bundle sheath cells, the chloroplasts which are present are agranule type. That means they will help only in dark reaction. So here in the chloroplast, that is in the stroma, there is this RUBP which will bind with this carbon dioxide in presence of Rubisco. Now in this situation, what is or what are the conditions for Rubisco? Where is oxygen released? Where light reaction takes place, in the mesophyll cell. So oxygen concentration definitely is not higher here. Secondly, the site of C3 cycle from upper cells has been shifted to lower cells, so the temperature is lower. Both conditions where the behavior of Rubisco changes have been taken care of. So now this complete cycle will be C3 cycle, normal, and this will result in formation of carbohydrate, that is glucose. So the problem which was due to the changed behavior of Rubisco has been taken care of. Now this pyruvate will go back or will diffuse into mesophyll cells. This pyruvate is a 3-carbon compound, and we have to regenerate this phosphoenol pyruvate. So this pyruvate has come here. This pyruvic acid is without phosphate. So the phosphate has to be added in order to regenerate phosphoenol pyruvate. So in this case, ATP is required. This ATP dissociates into AMP, that is monophosphate, from triphosphate to monophosphate. So we can say that one ATP dissociating into one AMP, that is one phosphate, second phosphate, third phosphate, it is changing into adenosine monophosphate. That means these two bonds are broken. Two bonds are broken. So energy which is released from these two bonds is required for this phosphorylation. So whether we call it one ATP getting dissociated into one AMP, or we can say it is equivalent to two ATPs dissociating into two ADPs. See, this is one situation where one ATP is getting dissociated into one AMP. How many bonds are broken? Two phosphate bonds. If I write two ATPs dissociate into two ADPs, how many bonds are broken? Again, this is one phosphorus, second, third. This is another ATP with three phosphates. So this and this, again two bonds are broken and the energy is required. So one ATP getting dissociated into one AMP is equal to 2 ATP. So in order to convert carbon dioxide into glucose, that means 6 carbon dioxides are required. For every carbon, 2 extra ATPs are required. We have already discussed that in C3 cycle, in C3 the ATP required are 18 per glucose molecule. To synthesize one glucose molecule, that means to fix 6 carbons, 18 ATPs are required. And in this C4 cycle, that means we have to include this as well this. So in C4, C3 anyways is taking place plus this extra. So 1 carbon, 2 ATPs, 6 carbons, 12 ATPs. So in C4 cycle, 12 ATPs plus 18 ATP requirement due to C3 cycle, this much is required. C3 cycle anyways is taking place, this will require 18 ATPs, 12 extra. So total energy requirement is going to be 30 ATPs in case of C4 cycle. But by spending some extra energy, the plant is able to fix carbon in the form of carbohydrate. So this, okay, one more additional change here, it is again oxidation reduction reaction, that is NADP will change into NADPH2. So this reaction and this both are redox reaction, oxidation reduction reactions. And this is important, which we have to remember, that one ATP changing into AMP is actually equivalent to 2 ATPs. That's why we have counted 12 ATPs. So by this changed position of T3 cycle, that means instead of mesophyll cells, shifting it to bundle sheath cells, and second, that is the enzyme which accepts carbon dioxide in mesophyll cells is pepco, whose behavior or activity does not change because of oxygen concentration or temperature concentration. So C4 plants, by showing some adaptations like Krenz anatomy and dimorphic chloroplasts, have adapted themselves to overcome all the problems which can exist due to photorespiration. So we can say that C4 plants do not undergo photorespiration. So this is one category of plants. They have avoided photorespiration problems. The second category of plants, they are called CAM plants. They have also undergone some changes to overcome these problems. Let us talk about CAM plants now. One more category of plants which have adapted themselves to overcome these problems of photorespiration, they are called CAM plants. CAM stands for crassulacean acid metabolism. Plants which show crassulacean acid metabolism, they are called CAM plants. Examples, the plants which show this type of metabolism, it includes flowering, xerophytes, like succulent plants, opuntia type, members of Crassulaceae family, and some members of Euphorbiaceae family. Some members, most of the members of Euphorbia and all Euphorbiaceae family, they show C4, but some members they show CAM metabolism or CAM type of pathway also. In this, the change is not the site, the change is the time. In them, the stomata open during night. Such plants where stomata open during night or such stomata which open during night, they are called scotoactive stomata. So they have stomata which open during night. So now let us take the situation. Say this is the epidermis and here are the stomata and we are showing it open, that means this is during night time. This is what happens during night, that is stomata are open. And let us draw one cell here which is representing the mesophyll cell. So here chloroplast, everything will be in mesophyll, no change of site like C4 plants. So this is mesophyll cell. Let us also draw another situation which we would see during daytime. So again the epidermal cells, and here let us make the stomata which is closed. So this is during daytime. Stomata are closed. And we will see what happens in the same mesophyll cell during daytime. During night, when the stomata are open, gaseous exchange can take place. So carbon dioxide diffuses into the cell. gaseous exchange is possible. So there can be gaseous exchange for respiration, it can also take place for photosynthesis. So carbon dioxide is taken in. This carbon dioxide is accepted by phosphoenol pyruvate, the same 3 carbon compound which we saw in case of C4 plants. So it is accepted by phosphoenol pyruvate. The enzyme is going to be PEP carboxylase. So it will help in carboxylation of phosphoenol pyruvate, and the first stable compound will be oxaloacetic acid, same as C4 plants. This oxaloacetic acid is converted into malate, same reduction reaction, that is NADPH2 will get oxidized into NADP and oxaloacetate will get reduced into malate, and this malate gets stored in the vacuole. So if we draw one small vacuole here, this malate diffuses in and it is stored. So what is happening during the entire night is carbon dioxide is taken in, accepted by phosphoenol pyruvate, converted into malate, and that malate is stored. So basically they are just taking carbon dioxide and converting it into malate and storing it. All night the same thing happens. During the entire night this PEP supply has to be maintained. So this PEP supply is maintained from chloroplast. So from chloroplast, this starch, it dissociates into a triose sugar, and this triose sugar keeps regenerating PEP, so that during the entire night when stomata are open, carbon dioxide is diffusing in, there should be phosphoenol pyruvate to accept it. So in case if this number is less from starch, it can be regenerated. This is going to take place during night time. During day, the stomata are closed, that means there is no exchange of gases. Exactly reverse situation takes place during daytime. That means from these vacuoles where this malate was stored, so this malate will diffuse out and it will, which is malate is a 4 carbon compound. So this 4 carbon compound will dissociate into 1 carbon in the form of carbon dioxide and 3 carbons in the form of phosphoenol pyruvate. Again for this oxidation reduction, so NADP, or let us put the arrow like this, NADP will get into NADPH2. So malate, which was stored in the vacuole throughout the night, diffusing out, changing into a 3 carbon compound, phosphoenol pyruvate and carbon dioxide. At this time stomata are closed and this carbon dioxide is accepted by RuBP in presence of same rubisco, and there will be C3 cycle. This will take place in chloroplast. So in chloroplast C3 cycle will take place, which results into formation of glucose. Just reverse what was happening during night, exactly reverse reaction takes place here. What happened during night? Because the stomata were open, carbon dioxide was taken in, accepted by a 3 carbon compound called phosphoenol pyruvate, which changes into a C4 compound, oxaloacetate, converted into malate by reduction, malate stored all night. During day, stomata are closed, no gaseous exchange, neither oxygen nor carbon dioxide. Malate diffuses dissociates into one 3-carbon compound and 1 carbon is released in the form of carbon dioxide. So, oxygen concentration is not there because gaseous exchange is not taking place and carbon dioxide is released. So, compared to any other gas, carbon dioxide concentration is more. So, RUBP will accept it and the enzyme which is going to help is Rubisco. It will help in carboxylation. C3 cycle will take place. Glucose will be synthesized. So, these CAM plants, they have avoided photorespiration by changing the time. During night, take in carbon dioxide, store it. During daytime, release that carbon dioxide and perform dark reaction, that is C3 cycle. So, C3 cycle here is taking place in daytime, though we call it dark reaction as it is independent of light, but still the time is that it is taking place during daytime. And the carbon dioxide which is released here is actually not coming right now during daytime, it was taken during night and was stored in the form of malate. So, C4 plants and CAM, they have adapted themselves to overcome the problem of photorespiration. C3 plants are normal, but whenever the temperature is higher, whenever oxygen concentration is higher, then Rubisco starts acting as oxygenase. These two categories, C4 and CAM, these plants, they have adapted themselves to avoid this photorespiration problem.